So my name is Boa Kim. I'm an assistant professor at the University of North Carolina. So the project that I'm going to tell you today is about endothelial cell lipid droplet metabolism and how it may link metabolic syndrome to blood pressure elevation. First, let me start with reminding you that metabolic syndromes like these are a big problem in the world. They are the major threat to global health, and nearly half of the adults in the United States have these metabolic syndromes. And these things are oftentimes interconnected, so they are interacting each other in a way that is poorly understood. But one of the things that happens with high-fat diet and obesity as part of the metabolic syndrome is that you develop hypertension via a mechanism that we don't fully understand. But there are quite a bit of literature that suggests that fat uptake increases your blood pressure. So this is just one example that just acute infusion of intralipid into people fairly quickly, within just a few hours, increases blood pressure in humans. And we see the same thing in mice. This is a study that we've done ourselves, where we have wild-type animals that have implanted telemetry device to measure blood pressure. And if you give them high-fat diet, their blood pressure goes up. So we are interested in this question of how does this high-fat diet and hyperlipidemic state contribute to hypertension? And last background slide is to remind you that hypertension is a very complex process of blood pressure regulation. But obviously, it's regulated at the level of endothelial cells because they continuously release vasodilators, including nitric oxide, NO, which is the potent vasodilator, which was a Nobel Prize in 1998, that speaks to the smooth muscle cell to modulate vasodilation and blood pressure. And this nitric oxide is generated by the enzyme called eNOS, endothelial cell-specific anosynthase enzyme. So the question we had was, what does a high-fat environment do to the endothelium? And first thing to say is that the excess fat forms lipid droplets in endothelial cells, which was a discovery from Bill Sasser's lab that was published just several years ago. And in this really nice study, they show the presence of lipid droplets in the endothelium for the first time in vivo. So this is just our version of the very same experiment that they showed in their paper. So when you take wild-type animals and you give it an olive oil by gavage, and after 3 hours, open up the aorta and alpha stain with BODP, which is a dye that stains neutral lipid in green. So all these green stuffs are lipid droplets. And we co-stain with CD31, the endothelial cell marker. You can see a whole bunch of lipid droplets are accumulated inside the endothelium of the aorta. And here is a lipid droplet contents that's quantified. And we found that it's not just a ton of olive oil gavage that does this. So here's an experiment where we simply put the animals on an ad-lib high-fat diet. And we found that just this one night of high-fat ad-lib consumption can also cause the lipid droplet accumulation in the endothelium. And what was interesting from this really nice work by Bill Sasser's lab was that these lipid droplets are bona fide lipid droplets, and they follow the same biochemistry that's studied in adipocytes. So here's an experiment again from us showing that their finding is true. So when you take endothelial cells, you don't see much of lipid droplets. But when you knock down ATGL, which is the enzyme that breaks the lipid droplet down, then you see these lipid droplets accumulate. And if you double knock down DGAT, which is the enzyme that produces the triglyceride, then these lipid droplets go away. So you can do this in cells. You can also do this in ex vivo cultured aorta. So it's very clear that the biochemistry is the same. So it was all in cell culture, and we were very interested to see whether this is also going to be true in vivo. So with absence of ATGL, for example, give you an accumulation of lipid droplets? And if so, what would be the physiological or pathophysiological consequences of having lipid droplets in the endothelium, where we don't usually think about they are? So we generated endothelial cell-specific ATGL knockout animals, and as you can see, very efficient deletion of ATGL in the isolated endothelial cells. And the outcome was exactly as we predicted, which is that a lot of lipid droplets accumulate in endothelial cells, and they do so throughout the body. So this is a Holman staining of solus. The muscle fibers are running this direction, and capillary vessels running on and between fibers are stained with CD31 in red. And you can see the lipid droplets, the green dots are lining all the vessels in the knockout animals. And of course, the next question becomes, what is the phenotypic consequences in these animals? And I can tell you that at first, the mice were completely normal. Their body weight was normal, body composition was normal, plasma, everything was normal. Their response to glucose tolerance test and response to oral fat tolerance test was normal. And pretty much the only thing that wasn't normal in these animals was that their blood pressure. So these knockout animals have elevated blood pressure compared to the wild-type control mice. So back to this statement about endothelium regulating blood pressure through nitric oxide and enos, quite strikingly and surprisingly, it turns out that in these animals in which the only modifications that we've done to these mice is to get rid of ATGL in the endothelium, they drop their enos content. So here's enos mRNA expression in isolated endothelial cells, and here is ANFA's picture of the aorta. So enos is in green in this case. So if enos is down, then NO is going to be down. And again, enos is endothelial NO synthase enzyme, which generates nitric oxide. But nitric oxide is very unstable and it's hard to measure, but there are byproducts of the presence of the nitric oxide, which we can measure, such as plasma nitrate and nitrite, they are down, and urine nitrate and nitrite, they are down in the knockout animals. So clearly, NO is down in these animals. So if NO is down, presumably the vasodilation should be down, and that's exactly what we found. So we can take the carotid artery from these animals and explant them in this cool pressure myography apparatus, and we first maximally constrict them with phenylephrine, and you give vasodilators such as acetylcholine, the vasodilator that requires nitric oxide to work. So in other words, acetylcholine talks to the endothelium, says secrete NO, and NO talks to the smooth muscle cell and relaxes vessel. And you can see that it takes quite a bit more of acetylcholine to get these vessels from the knockout animals in blue line to relax completely, which is a typical response for insufficient NO production for endothelial cells. So this data raised in our mind the notion that perhaps the high-fat state leading to hypertension might be via the lipid droplets in the endothelium, and it occurs via the reduction of enos, NO production, and impaired vasodilation. Then next question is, how does this work? So we tested whether it's the lipid droplets that's dropping enos in the endothelial cells by getting rid of the lipid droplets in the ATGL knockdown endothelial cells. So there are two ways to get rid of the TG particles, and one of them is to enhance lipolysis. So I used forskolin that activates adenylate cyclase and increases TG hydrolysis and therefore makes lipid droplets go away, and found that it rescues enos expression. So yes, breakdown of lipid droplets does rescue enos expression. And the other way to get rid of the TG particle is to prevent their synthesis pathway. So I blocked DGAT, the final step in TG synthesis pathway, converting diacylglycerol to trisylglycerol by using an inhibitor, and therefore removes lipid droplets in SITGL endothelial cells. And we found that it rescues enos level. And consistent with this drug treatment, double knockdown of DGAT with siRNA showed the same rescue effect, and targeting ACSL further upstream in TG synthesis pathway also rescued enos expression. So is it the lipid droplets that's reducing enos? So yes, we are pretty convinced that it's the lipid droplets, not something else that ATGL is doing to these animals. Then next question is, how do these lipid droplets suppress enos level? So we went back and thought about whether the enos suppression is at the transcriptional or at the post-translational level. And we've ruled out the possibility of post-translational regulation because one, we see less of enos mRNA transcript with qPCR. And also based on this MG132 experiment that, which shows that the enos suppression is at least not due to the proteasome-mediated degradation of enos protein. And another possibility that we tested is post-transcriptional suppression of enos. So it is possible that the enos mRNA gets transcribed normally, but if somehow it gets destabilized and degraded, you detect less of enos mRNA with qPCR. So speaking of enos mRNA stability, it is reported to be highly stable in endothelial cells, and with actinomycin D treatment, which inhibits RNA polymerase and blocks transcription, you can measure over time how much of the mRNA of your interest is remained in your cell, which indicates how stable your mRNA of your interest is. And here's my version of the ENOS mRNA stability assay, and consistent with what's reported in the literature, in SI control, the ENOS mRNA was highly stable and didn't degrade over a day. And when you knock down the TGL, you can see that their ENOS mRNA gets destabilized and degrades significantly more than SI control cells. So now we have ENOS mRNA destabilization in our model as a potential mechanism. Then next question is, who does this? Who is the destabilizer of ENOS mRNA? And from literature search, we found that there are 3 interesting candidates that may regulate ENOS mRNA stability, EEF1A1, PTBP1, and PCBP1. And all 3 of these proteins are reported to directly bind to ENOS mRNA 3' ETR region. So these are interesting candidates to follow up. The next question is, do they have anything to do with lipid droplets? So in an effort to figure out the mechanism of ENOS suppression, we've performed endothelial lipid droplet purification for proteomics analysis with mass spec. And before sending these samples out for proteomics and mass spec analysis, we validated whether what we have is really a pure lipid droplet fraction and whether there's any contamination by other intracellular organelles. And as you can see, ATGL and PLIN2, the well-known lipid droplet proteins, are there, but no contamination by nuclear proteins, cytosolic, and plasma membrane, Golgi, ER, or mitochondrial proteins were detected in this purified lipid droplets. So we sent this pure endothelial lipid droplet fraction for mass spec and got a list of a few hundred proteins. And interestingly, the gene ontology analysis of these endothelial lipid droplet proteins pointed mRNA stability regulation as a top hit. And what was even more exciting to us was that back to the ENOS mRNA stability regulators, EF1A1 was here, PTBP1 was here, and PCBP1 was here. So all 3 proteins that were known to regulate ENOS mRNA stability were identified in this lipid droplet proteomics analysis in endothelial cells. But of course, you don't trust your mass spec data until you validate it. So today I'm going to show you just a very brief validation of just one of the 3 regulators, PCBP1. So here is a cell staining of PCBP1 in red, and in the ASI-TGL cells, you can see PCBP1 localizes to the lipid droplets as shown with co-staining of ODP, the lipid droplet marker. And strikingly, PCBP1 knockdown, double knockdown rescued ENOS level. So putting this data together, what we think potentially might be happening is high lipid state is causing accumulation of lipid droplets in the endothelium, and that destabilizes ENOS mRNA, maybe via the localization of PCBP1 to lipid droplets, and the destabilization of ENOS mRNA leads to reduced ENOS protein, reduced NO production, and impaired vasodilatory function, and elevates your blood pressure. So if our model is true, then blocking endothelial lipid droplet accumulation in this high-fat context, it should also prevent the blood pressure elevation by a high-fat diet. So the last data to show you is back to wild-type animals. So as I showed you in an earlier slide, on high-fat diet, you get accumulation of lipid droplets in various different vascular beds. And if you give DGAT inhibitor during high-fat diet to these wild-type animals, then these lipid droplets go away. And strikingly, we found that the blood pressure elevation by high-fat and high-salt diet challenge also gets alleviated. And finally, we examined the EQTLs of lipid droplet metabolism enzymes in the vasculature and their relationship with blood pressure in human cohorts in collaboration with Dr. Lavina Penn. And he identified that there's one SNP that is associated with blood pressure among all the SNPs of various enzymes in lipid droplet metabolism pathway. And that SNP was on PNPLA2 gene, which is the gene that encodes ATGL. So what this data is telling us is the higher the ATGL is expressed in your vasculature, which is the enzyme that breaks down lipid droplets and therefore makes your lipid droplets go away from your endothelial cells, the lower your blood pressure is. supporting our model on the causal relationship of lipid droplet accumulation and blood pressure elevation. So back to this picture from the very first slide, as a proof of concept, we think that the excess lipid accumulation in endothelial cells, at least in part, contribute to this picture on how hyperlipidemic state leads to blood pressure elevation. And the last point to make is that lipid droplets are really cool intracellular organelle, and they used to be an inert thing. It has been thought of as a storage place to store excess fat in the cells, but increasing number of evidences have demonstrated that they do more than just storing fats. And in fact, they are really dynamic organelles carrying out various functions, including sensing and responding to metabolism, innate immunity or aging, and etc. And we believe this study is providing another example of that. This is my currently small but growing team at the University of North Carolina. Thank you for tuning in, and I'd be happy to take any questions.